so I think that this would be part 3 for you guys. And there's, again, feel free to look at whoever's lecture. I don't record all of them, and sometimes I say different things, sometimes I forget things. So anyway, if you're wanting to review a lecture, try the other ones too. They also won't always end in the exact same spots, but you could fast forward or do whatever. Okay, so we were on lipids. The first big group of lipids we already talked about. What am I talking about? Fats and oils. They were the triglycerides, if you remember. They had 3 fatty chains to a glycerol, okay? I don't expect you to memorize every detail, so I'll make sure I prep you for the test, but I want you to think about it right now. Reach beyond what I'll test you on. There's 2 more groups here. There's phospholipids and cholesterols. These are steroids. This is the most diversified organic macromolecule group there is. Remember we talked about carbohydrates? This one has the most variety in it, okay? Proteins are crazy too, we'll get to them. But these have a lot of different monomers, a lot of different building blocks and stuff. Okay, so phospholipids first, because these are both very different. Phospholipids, I'm going to show you a picture. They have the glycerol, but only 2 fatty acid chains, and they have this phosphate group. I'll explain in a little bit. And then the phospholipids are in every single one of our cell membranes for all life. Cell membranes are made out of phospholipids. They also have proteins embedded in them. So there's the simplest look at a cell membrane. See the phospholipid, the 2 fatty acid tails, and that head region is your glycerol. Now what's cool is this side doesn't like something. Do you see how they're lined up together here? But the head side of the molecule likes something. What am I talking about? The heads like something. What do they like? Look how they line up. Yeah, they like to be around water. What do you think about the tails? They are hydrophobic. So it says it right here, that is a weird thing. That's weird. Because that is hydrophilic, and that's supposed to say hydrophobic. Goofy. Fix that. This should say phobic, and that one's right, right? Okay. Well, make sure, I want you to label it right now. Tails are hydro-what? Phobic. They want to stay away from the water. That's why they line up like that. The heads and the tails are not bonded together, okay? They're free moving. We'll talk about them more later. The heads, like the water, hydrophilic. Now, cholesterol, their structure, form, and structure are very important again. And I'm going to talk to you about it more later, okay? But that's the other type of lipid. You should know that cholesterols are lipids. And you kind of think about that, right? Don't they say if you eat a lot of fat, you get a lot of cholesterol in your arteries? You guys, right? So it clogs up. And that's because it's kind of this kinky shape, okay? And over here in our phospholipid cellular membrane, see, here's the picture of the cholesterol, okay? Now, cholesterol is really cool, and we'll get to cell membranes later in detail. Right now, I just want you to know that lipids are the main component of cell membranes. Okay, now we get to the crazy proteins. So every living cell contains from several hundred to several thousand different macromolecules known as proteins. So here I told you that the lipids were more diverse because you had 3 very different types of lipids, and then you even had 2 different of the fats and oils, the saturated and unsaturated. So there's only like 3 or 4 different types, but they're very different. Here you have tons of proteins, They're all built sort of the same, but they're varying shapes and sizes. They're built of the same repetitive monomer, which is the building blocks, which is amino acids. But then they're folded, and that's what we're going to do some cool stuff with our models in a minute. So the proteins, there are a ton of them, and they do just a huge variety of different things. That's their function. For example, we have structural proteins. We have hemoglobin is a protein in our red blood cells that's attracted to oxygen. Messengers, we have little receptors. Remember, back to that membrane, didn't I say there were proteins in the membrane itself? Protein channels. Here's a protein with a carbohydrate chain. That's for receiving messages. You have all different proteins. I'm not even down my list here. Enzymes are awesome. Okay, they help reactions happen. We'll be working with some enzymes in another chapter or two. This is amylase. That's in your spit. Right now, your saliva, it breaks down carbohydrates. We'll do a lab on that. Okay, now, the monomers. Back to carbohydrates. Those monosaccharides. Remember glucose and fructose? This was the simple sugar. Those are the monomers of the disaccharides and the polysaccharides. Like you could get glucoses stringing together. So you see how the glucose is the monomer of a big starch molecule. Then we had the fats, and that one was that diverse group. So we had fatty acids as part of the monomers, and this glycerol as part of the building blocks, and that's why it's so diverse. Now the building blocks of proteins are what? What's it say? Amino acids. You've got to start memorizing that right now. It is a theme that we will have all year long. So the building blocks of proteins are what again? Amino acids. There's 20 different ones. They look very, very similar to each other. They look like this. They have this group over here. They have this group over here. They have a C and a H, and this is where it varies. And that changes, and that's why we have 20 different amino acids. And those amino acids strung together make those crazy proteins. Okay, those big globular weird things. Okay, now, they're held together with covalent bonds, and I don't know, just because we like to talk and name everything, we decided we'd name those covalent bonds that hold amino acids together, what? Peptide bonds. I'm not going to over worry about it, but because we call those covalent bonds peptide bonds when they're bonding amino acids together, then we have a polypeptide, which is a chain of amino acids. It's very wordy. Don't get too caught up on it right now, but that's what we're talking about. Let's check this out.
A peptide bond forms when the acid group of one amino acid joins to the amino group of another amino acid. In this example, the acid group of glycine joins with the amino group of alanine. The result is glycoalanine. The acid group of glycine loses an atom of hydrogen and an atom of oxygen. The amino group of alanine loses an atom of hydrogen. These atoms combine to form a water molecule. More peptide bonds can form in the same way, creating a chain of amino acids called a polypeptide.
Okay, so you can see here's a glycine. It's going to have a different variable group up there than this one, and they actually gave it to us on this one because it's simple. It's just that CH3. They could fit it there for us. And just like the other reactions of bonding the glucoses together, we lost some water again. Okay, so whoops, that was too fast, right? So those amino acids all link up together and form a chain. Most of the time that chain needs to be folded into a particular shape, okay? So these shapes of proteins are so complex, we decided to organize them. If it is just a simple chain and it doesn't have to be folded, the protein is known as having primary structure. You see that at the top? Now, secondary structure of a protein has it, and this is you're going to find these models. You're going to play with this, so make sure you're hearing me. Okay, secondary structure means two things. The chain has been pleated back and forth, or it has been spiraled in a helix. What type of structure is that called? Secondary structure. So a helix or a beta sheet. And these are still very simple for proteins. But now check out this one. This one's getting a little different. This is tertiary, meaning the third type of structure. We're getting more and more complex. And now what you do is you have interactions between those variable groups on those amino acids. Let me see what else I have here. Do I have a better picture? Darn it. Is there more on that slide? Okay, so what I'm going to show you some more, but that variable group, which is also known as a side chain on that amino acid too, or they call it an R group because it's just that variable group, has its own properties. Maybe it's hydrophilic, maybe it's hydrophobic, maybe it's positively charged, maybe it's negatively charged. So these little side chains that are sticking out of the polypeptide chain might want to interact with each other, might be attracted to each other, so it starts folding up by these different attractions. Or maybe it's repelling and it's spacing out. That's when it starts having the interactions between the side chains. That is not a random thing. It is folded by those R interactions, those variable group interactions. And it'll stay like that because those are weak bonds holding it together. Okay? Then, quaternary, which is the fourth structure, is when we have more than one polypeptide chain. So now you see the color coding. We got purple, green, blue, and brown. Now you have 4 different folded up chains that are fitting together because of those weak interactions again, because of those attractions. Okay? Now since all these interactions of how the protein is shaped are weak, do you think it's easy for the protein to kind of fall apart if it's got in a bad environment, for example? And that's what happens when your meat is a protein that you guys eat. It looks all pretty and fleshy and red, right, when it's raw. And then you mean people heat it up, and does it change what it looks like? Because you've broken all these weak interactions that were making all those molecules work perfectly and hold their shape together. And you heated it up, and it falls apart. It gets denser. It gets stuck. It just changes its shape. Will it work anymore? No. Okay, it's not going to, for a while, you'll see, we'll be working with some raw liver. It'll still do cool reactions and stuff until you guys possibly hurt those structures. Okay, one more group. Oh, I'm so excited. This is almost over for you guys. I thought it was going to be a long one. One more group. Okay, let me review. We started with what type of macromolecules last class? Before lipids? Carbohydrates, then we move to lipids. I just talked to you about proteins, and the last thing is awesome. Okay, the NA. You maybe don't, that might not sound familiar, nucleic acid, but you are very familiar with it. What does DNA stand for? Nucleic acid, DNA, and RNA, ribonucleic acid, nucleic acids, okay? So then, what we're talking about here is DNA and RNA. So we're not talking about sugars, we're not talking about proteins, we're not talking about fats and lipids, we're talking about DNA and RNA. That there's only 2 types of nucleic acids. Their monomer is a nucleotide. I'll show it to you in a minute. The nucleotide has a sugar on it, and it has one of 5 nitrogenous bases. So I said there's 20 different amino acid monomers. How many nucleotides are there? 5. Hold on. Oh, there's also the phosphate group. Okay, so here is a picture of the 2 sugars. DNA sugar, deoxyribose, is a little bit different, having to do with the oxygen. D, like degrease, to get rid of. D, you know what I mean? 1 less oxygen. Ribose has another oxygen. This is the sugar in RNA. The other one's the sugar in DNA. These are 4 nitrogenous bases out of the 5. DNA only has these 4. And that is stuck onto the sugar. RNA, there's some difference here. RNA is not double-sided, it's single-sided. We'll talk a lot more about RNA in the future. The other thing I want to make sure you know, though, from this lecture is that it has the 5th one. It has 3 of the same and 1 different one. I'll show you in a minute. So first, back to DNA and these bases. Do you see the shapes? Adenine, because we're making 2 sides for DNA, the double helix, right? So A fits with what? So they bond. C with G. Now these are models, they're not exact, but when you saw the formula configuration back here, you could see, okay, here this one has these 2 H's right here. It's going to fit with a G. See how that is going to fit right in between that? See, now these ones are a little bit different. This is more rounded. This is more sticking out. This configuration can fit right in here. But anyway, that's a little more involved. So, those are our nucleic acids. Our monomers are what? Nucleotides. We have A's, T's, C's, and G's. That's adenine, thymine, cytosine, guanine. And the 5th one is, instead of thymine, RNA's 4 different nucleotide bases is uracil. And it's the same shape as the thymine. Yes, the 5, yes. No, you're not, you don't have, we'll all help you more with that. Okay, so Watson and Crick get most of the credit for coming up with the structure of DNA. However, there's some controversy, and they definitely had some help from Rosalind Franklin. Now, Wilkins was her assistant, and Wilkins actually was the one that shared a very important X-ray photograph she had taken that allowed Watson and Crick to realize it was a spiral. So there's some controversy. There was some definite sexism thing going on too, because she was a pioneer as a female and a long time ago, and that was being frowned upon, right? Not anymore. You know, scientists, female scientists are outnumbering male scientists right now. Did you guys realize that? Especially in more of the life science fields compared to the engineering and physics, even though it's getting very equal all over the place. Okay, so that just goes over more of what we're talking about. The double helix, let's listen to this, and we're almost done.
The DNA double helix is composed of 2 long chains of nucleotides. The backbone of each chain, shown as ribbons, is composed of alternating sugar and phosphate molecules that connect the nucleotides. The 2 chains are held together by specific patterns of hydrogen bonding between pairs of bases, A to T and G to C, and the chains coil around a central axis to form a double helix. The diameter of the double helix is equal along its length because a double ring A or G base called purines always pairs with a single ring T or C base called pyrimidines. There are 10 nucleotides per turn of the helix. Only a small portion of the model of a DNA molecule is shown in this example.
So you would ask a question about what you need to know for the test for DNA. For example, you would need to be able to label something like this. You would need to know that A's bonds to T's, that C's bond to G. You need to know it's a double helix. You need to know that the backbone is alternating phosphates and sugars. And that's basically it. And then RNA, single-stranded uracil instead of thymine. The 2 sugars in each of them, just that RNA is ribose and DNA is deoxyribose. And that these are nucleic acids, and they're built of what monomer? Nucleotides, right? And that was very fast, I know, but let me stop.